Hello everyone. Today we'll be going over regulatory T cell isolation using the 18063 kit on the EasySep Magnet. Performing magnetic cell isolation of regulatory T cells, also called Tregs, involves first isolating CD25 cells via a column-free immunomagnetic pod selection for CD25 using the releasable RapidSpheres. Then, these bound magnetic particles are removed from the isolated 25 cells such that in the next step, the unwanted non-Tregs are targeted for depletion. Consequently, the final isolated fraction has highly purified regulatory T cells that have high levels of FOXP3 expression and are immediately ready for downstream applications. As well, at the very end of this video, we will also demonstrate the optional steps necessary for the isolation of CD4 positive 25 negative responder cells that can also sometimes be desired for use in functional studies. I did want to outline an important consideration, which is when you're using the 18063 human regulatory T cell kit with the EasySep Magnet, to ensure to contact STEMCELL via tech support at stemcell.com or certainly via your individual sales representative and request 2 additional vials of catalog number 50100. These are the extra Dextran RapidSpheres, which will be needed when you're using the EasySep Magnet. For using the 18063 kit with the EasySep Magnet, you will need the following things, including your prepared cells, which should be at the cell concentration of 50 million nucleated cells per mil. In this case, we have 20 mils, as well as 50 mil Falcon tubes, which are compatible with the EasySep Magnet. You will also want to have the EasySep buffer, serological pipettes, 25 mil sized or 10 mil sized, the serological pipette controller. You're going to want to have the extra Dextran RapidSpheres, as well as P1000, P200, and associated tips, timer, as well as liquid waste container and waste bag. Note that all the following steps will be done at room temperature. First, the starting cell sample will need to be prepared, and we do have other technical resources to detail the necessary steps depending on your starting cell sample type. Note that the 18063 kit can start with peripheral blood mononuclear cells, or PBMCs, from whole blood subjected to a density spin, as well as an appropriately processed fresh leukapheresis collection. Additionally, it can be possible to start from cryopreserved PBMCs, though you will want to ensure that these are appropriately prepared, such as using DNAs. But an important consideration is that the performance may not be as ideal using cryopreserved PBMCs compared to starting with freshly prepared PBMCs. Once the starting cells are processed, a cell count will be conducted. Normally, we suggest executing a manual nucleated cell count using an aliquot of the cells diluted with 3% acetic acid with methylene blue and loading this onto a hemocytometer for manual counting. Once the nucleated cell concentration is obtained, adjust the volume so that the starting cell concentration is 50 million nucleated cells per mil as prescribed on the product information sheet. Once that cell concentration is achieved, evaluate how much volume you have, as this will be used to select which magnet is appropriate. In our case, we have 1 billion cells, which means that this is in a 20 mil volume, and thus the EasySep magnet is appropriate for this isolation. Now that the starting cell volume is in the appropriate tube and all the reagents are available, the isolation can commence by first adding the C25 pod selection cocktail at 50 microliters per mil of cells. In this case, since we have 20 mil starting cell volume, we will add a total of 1,000 microliters of the CD25 pod selection cocktail. When adding this volume, you're going to want to ensure that the tip goes into the cell suspension, and then that is followed by a quick trituration. So here, just in the cell volume, dispense, and pipetting up and down. Having added that, I personally also like to suggest to also do a mix. Once you have mixed the cells, you can cap the tube again and commence a 5-minute room temperature incubation. Take note that this kit uses two types of magnetic particles. You have the releasable particles, which is catalog number 50201 and are used early on in the protocol, as well as the Dextran RapidSpheres with catalog number 50100, which are used later in the protocol. Therefore, for this next stage, it will be the releasable RapidSpheres 50201s that will be used first. However, before these particles are used, it is very important to take this opportunity while the cocktail incubation elapses to vortex these particles for at least 30 seconds. As well, in addition to the releasable RapidSpheres, the next step will also require use of the CD4 T-cell enrichment cocktail, so it is important to have this readily at hand. Once the CD25 cocktail incubation has elapsed, first add the releasable RapidSpheres at 30 microliters per mil of cells to the sample. In this case, since we have 20 mils of cell volume, we will be adding 600 microliters total of these releasable RapidSpheres. Again, when you're adding these releasable RapidSpheres to the cell volume, do ensure to put the end of the tip into the suspension, dispense, and then just triturate up and down a few times. Afterwards, you're going to want to take a pipetter and just pipette that up and down to mix. Immediately after mixing the sample, proceed to add the CD4 T-cell enrichment cocktail. This will be added at 50 microliters per mil of cells. Take care to add the cocktail directly into the cell suspension. Since we have 20 mils and it is prescribed to be added at 50 microliters per mil, we are adding 1,000 microliters total. So you want to dispense that in and then just triturate up and down. Followed by a quick mix. After the mix, you may cap the tube, set your timer for the 5 minutes, and let that elapse at room temperature. Once the incubation time has elapsed, add the recommended medium to top up the sample to the indicated volume prescribed on the information sheet. If the start sample volume had been 20 mils or less, then it would be topped up to 25 mils. Otherwise, if it is more than 20 mils start volume, then this will be topped up to 50 mils. In our case, we are starting with 20 mils of start sample volume, and so we want to take this up to 25 mils. However, we do need to keep in mind that we've already added some reagent volume. So in our case specifically, we will be adding 2.4 mils of this EasySep buffer. And after we add this, we will then do a mix. So that is why I am using a 25 mil serological pipette. Okay, we'll just add this in. Pipette up and down to mix. And then we will be placing this into the EasySep Magnet for what I like to call separation 1A. And when placing this into the EasySep Magnet, you will want to have it so that the graduations that are normally on the sides of the tube are facing inward. Once the 10-minute separation time has elapsed, you're going to want to aspirate the entire volume out of the tube and transfer this into a receiving 50 ml tube. Typically, it is advisable to try and aspirate this all out in one aspiration. Um, so you have to use an appropriately sized serological pipette. Some people may find that their pipette aid with a 50 ml pipette will drip. So in that case, I suppose it is okay to use a couple draws of a 25 ml serological pipette, though in this case, it should be okay. And you'll want to notice that as I am aspirating down, I am going from the top down, and I am ensuring to maintain the tip of the serological pipette forward. Okay, then I will transfer this to the receiving tube, and then I will just leave this in the magnet. Next, do remember that this kit uses two types of magnetic particles, the releasable RapidSpheres as well as the Dextran RapidSpheres. For this next step, you will be using the releasable RapidSpheres. However, as with before, you do want to ensure that these have a nice 30-second vortex before aspirating. Once vortexed, these releasable RapidSpheres will be added at 15 microliters per mil of the original start sample volume to the 50 ml tube containing the collected supernatant. Or put another way, you will be adding half of the volume of releasable RapidSpheres that had previously been added. In our case, we had previously added 600 microliters of these releasable RapidSpheres, so now we will be adding 300. Set this to 300. Okay. And as always, when adding your reagents to the cell volume, you want to ensure that the end of the tip is submerged, and then you triturate a few times, followed by doing a mix. Note that it is very important to do a very thorough mix at this stage. And as a matter of fact, what some people alternatively like to do is that ahead of having aspirated this supernatant, they would have actually have added the particles to the bottom of this receiving tube, and then to that have added the supernatant. You can try doing that, but certainly as long as you ensure to do a very thorough mix at this stage, you will be perfectly fine. Okay, then after you have mixed, you may set your timer for a nice 4-minute room temperature incubation. Once the 4-minute incubation time has elapsed, you're going to want to aspirate that volume back into the tube within the magnet. So again, select an appropriately sized pipette to aspirate out the entire volume. In this case, we could use a 25 ml pipette or a 50 ml, using a 25 here. And I will simply dispense this back in. Once that has been dispensed, I will set my timer for a 10-minute separation, which I like to call separation 1B at room temperature. Once the 10-minute separation time has elapsed, aspirate out the volume into a 50 ml receiving tube. Again, ensure to use an appropriately sized pipette so that the entire volume can be aspirated in one draw. And as noted previously, please do ensure that when you're aspirating to draw from the side furthest away from the magnet at the back, and then dispense this into the waiting 50 ml receiving tube. Ensure to save this receiving tube, particularly if you would like to obtain CD4 positive, CD25 negative responder T cells, since this will be the start sample material. From there, you're going to want to take out the tube out of the magnet, and you can see that the particles will be on the sides. From there, you will need to add EasySep buffer, and the amount to add will be prescribed on the information sheet, such that if you had started with 20 mils or less, then you'd be topping up to 25 mils. Otherwise, if you had started with 20 mils or more than 20 mils, then you'd be topping up to 50 mils. So in our case, we had 20 mils, so we are adding 25 mils, and pay special emphasis to thoroughly wash the sides of the tube. And then while washing the sides of the tube, do ensure to mix up and down. Okay, now that that is mixed, this can be placed back into the magnet, graduations away from you, and then this will be another 10-minute separation at room temperature. Once 10-minute separation time has elapsed, you're going to want to aspirate out the volume in the magnet. And again, you want to dispense this into the liquid waste container. As with before, you want to position this so that you are placing the end of the pipetter away from the back where the magnet is, and start aspirating down, going down with the front of the liquid. Dispense this into the liquid waste container. Next, you can take the tube out of the magnet, and you may see the particles on the side, which we will bring back into suspension by adding the EasySep buffer. As with before, since we had a 20 ml start, we will be adding 25 mils of the EasySep buffer. Okay. So just want to showcase how you really want to thoroughly get that back into suspension. And then once you have this back into suspension, Just give it a quick mix. And then this will be going for the fourth 10-minute separation at room temperature. Once the 10-minute fourth separation has elapsed, you're going to want to aspirate that volume into the liquid waste container. So again, be careful to aspirate going slowly down on the side away from the back of the magnet. Perfect. Cap that off. Take your tube out of the magnet. And then you're going to want to use the recommended medium, in this case EasySep buffer, to resuspend those cells back to the start sample volume. So you might recall we had 20 mils of initial start sample volume. So let me aspirate that out and then very thoroughly getting the cells back into suspension. As always, do try and ensure to get the sides of the wall. Okay. Now that we have our C25 positive cells back into suspension, we can cap our EasySep buffer. And the next step is to add the releasable buffer. So in this case, we will be adding this at 100 microliters per mil of cells. So since we have 20 mils, we will be adding 2,000 microliters. I'll get the pipette, set this to 1,000. Add the first 1,000 in there, triturate. Next, I will be adding the second release buffer to make up the total volume of 2,000 microliters. Once the release buffer is added, you may notice that the instructions do call to vigorously mix, and this is very important. So take an appropriately sized pipetter and do ensure to vigorously mix at least 5 times. You just want to make sure that that release buffer is thoroughly distributed amongst the cell suspension. Okay. Should be good. The added release buffer will help remove the releasable particles from the labeled cells. However, of course, there are other 25 expressing cells in PBMCs, so now we do need to add the CD127 high depletion cocktail, which is called to be added at 50 microliters per mil of cells. In this case, it will be 1,000 microliters since we have 20 mils of cells in there. As always, when adding reagents, you want to ensure to have the end of the tip in there, followed by a trituration and a mix using an appropriately sized pipetter. Note that together with the already cell-bound CD4 enrichment cocktail that was added much earlier in the process, the CD127 high depletion cocktail will allow us to further isolate the CD4 positive, CD27, 127 low cells fraction from the isolated 25 cells. After this mix is done, set the timer for a 5-minute room temperature incubation. Take note that this kit does use 2 types of particles. However, now that we are in the later stages, we will be using the Dextran RapidSpheres, catalog number 50100. However, as before, these RapidSpheres, as with the releasable RapidSpheres, do have to be vortex for 30 seconds to ensure that they are thoroughly mixed in the vial before aspirating. Okay. Now, once that the 5-minute incubation has completed, we will be adding the vortexed Dextran RapidSpheres, and these are called to be added at 20 microliters per mil of cells. So since we had 20 mils to start with, that means we will be adding these at 400 microliters total. Added with the end of the tip inserted, then triturate, followed by a nice mix. And then once mixed, these will get set aside for a 5-minute incubation at room temperature. Once the 5-minute incubation time has elapsed, add recommended medium to top up the sample to the indicated volume prescribed on the information sheet. If the start sample volume was 20 mils or less, then it will be topped up to 25 mils. Otherwise, if it was more than 20 mils, you'd be topping up to 50 mils. In our case, we had our 20 mil start. However, we have added volume via the reagent additions, such that right now all we really need to add is about 1.6 mils, and then follow that up with a nice mix. Next, after you've mixed this, the tube will be placed into the magnet. As always, graduations away from you, commencing a 10-minute room temperature separation. Once the final 10-minute separation time has elapsed, you're going to want to aspirate the now desired supernatant out of the tube and into a new receiving container. As always, you want to ensure to aspirate having the end of the pipette towards the front, going down slowly. Okay. And now your isolated regulatory T cells are ready for immediate downstream uses. At this point, you can elect to optionally, if needed, use the save supernatant to obtain the CD4 positive, CD25 negative responder cells, as will be showcased in the next part of this video. Having isolated the Tregs, the saved supernatant that was set aside earlier can be used to obtain your responder cells. In order to do so, you will have to use the Dextran RapidSpheres. Specifically, you need to add these at 180 microliters per mil of the original start volume. Naturally, you will want to vortex these for 30 seconds before aspirating and dispensing them into your cell suspension. Once the Dextran RapidSpheres have been mixed, you will then add these triturate up and down. Triturate up and down. Triturate up and down. Having added this, you will want to do a quick mix. And then set this aside for 5 minutes. Room temperature incubation. Once the 5-minute incubation time has elapsed, place the tube with the graduations away from you into the EC50 magnet, and then commence a 10-minute first separation without the cap at room temperature. Once the first 10-minute separation time has elapsed, you're going to want to aspirate out the supernatant into a magnet-compatible tube. As always, when aspirating from the EC50 magnet, you want to position the end of the pipetter away from the back towards the front, and then slowly aspirate out. Always ensure to try and have a size appropriate so you can do the full draw in one go. Dispense this into the new tube, followed by taking the old tube out. And now, in its place, place the new tube and commence the second separation, which will be for 10 minutes at room temperature. Once the final separation has elapsed, aspirate the supernatant into a final receiving tube. As always, aspirate drawing from the front and dispense this into the new tube. At this point, you now have isolated your regulatory T cells as well as your responder CD4 positive, CD25 negative cells. If you have any other questions, please contact us at technical support or certainly liaise with your individual field application scientist or sales rep. And happy sciencing.